I'm very glad to start this morning in our time session with Chirag Jain. Chirag is assistant professor at the Indian Institute of Science. He has published several papers in computational pangenomics, and I'm really excited to have him here. Before joining the Indian Institute of Science, he got his PhD at Georgia Tech, and he has been a postdoc at the National Institute of Health. So I see from the title that you are going to talk about the problem of genome inference in a pangenomic setting. So thanks again for joining us, and we're waiting for your talk.
Okay, thank Gianluca. So my name is Chirag Jain. I'm a faculty member at the Indian Institute of Science. So it's a pleasure to talk to you all today about our recent work on pangenome-based genome inference via integer programming. So let me get started. And before I get started, I can mention that my talk is not is short. So basically, that means that there is a lot of time to ask questions in the middle. So please feel free to interrupt me by directly speaking, and I'll stop and answer your question. Okay, so let's get started. So I think all of you have seen the overview of pangenome graphs already, but just for completeness, let me remind you that the graph-based representation of pangenomes is one of the compact ways to represent a large collection of human genomes. This is this representation is made possible because of recent advances in genome assembly, the ability to get complete haplotype resolved genomes, and as a result, these graph-based representations also allow us to capture the variation in the human populations. So as a result, it addresses the limitations of the conventional reference that we were using, which was a string-based data structure. Now, a graph-based representation offers these benefits that can be helpful for improving genomics accuracy. So there are a number of applications of pangenomes, and especially in the context of human genomics, they allow us to mitigate the reference bias, uh, allow us to enhance the disease association with studies, and of course, getting insights into population genetics and evolution. For those who are still learning about this topic, there are excellent review articles written on pangenome algorithms and pangenome applications. So I refer you to those articles for more information. Now, addressing these applications is also not easy because pangenome graphs are now complex compared to the string-based representation. So there are a number of algorithmic challenges. For example, building the pangenome graphs itself is not easy. You have a very long sequences coming from large number of individuals, and determining how to construct an appropriate graph such that the common sequences are collapsed into a single haplotype or into a single vertex, and you can spell the individual haplotypes as a path in the graph. That's not an easy problem to solve. Similarly, mapping reads to a pangenome graph is another challenge because now there is a lot of combinatorial possibilities that you have in the pangenome graph. And finally, calling variants, like even once we have the accurate read alignments, or we have the short read sequencing dataset or long read sequencing dataset, calling variants in the target sample using a pangenome reference is also challenging. So in our group, we have been interested in all of these problems, and we have contributed some algorithms for each of these directions. For today's talk, I'm going to focus on the genotyping problem, which is our most recent work that is going to appear in Recon conference. So the next few slides, I'll give you an overview of what we did in this work. So just as a background, let me just quickly remind you what genotyping means. So in case, suppose if your target genome is haploid, that is the ploidy is equal to 1. Then genotyping is simply the process of identifying if a known variant is present at a specified locus. So for example, a known variant would be SNP, insertion, deletion, or a large structural variation. The process is to identify whether that variant occurs at a specified locus or not. In the case of diploid genomes, then the genotyping problem is to identify whether a known variant is present at both chromosomal copies or just one chromosomal copy or none of the chromosomal copies. That is, we have to account for the fact that now the ploid is 2, so therefore the genotyping has to be done on both copies of the chromosome. So given this background, there are a number of approaches there for genotyping problem. This is a fundamental problem in genomics. So if we think of the solutions that involve the use of a linear reference, and let us say that you have sequenced the target sample using short reads, then we have two kinds of methods. That is, one is alignment based, and the second is alignment free. That is directly, for example, using K-man counts from short reads. Now, alignment-based methods would align these reads to the reference, and based on the consistency of the alignments and how whether it's a match or a mismatch, one would infer the genotype. And in the case of alignment-free method, you would use K-man counts to infer the appropriate genotype. In this scenario, one can also use haplotype panels. For example, if I have some reference panels of large number of individuals available already, I can use those panels for phasing as well as imputing missing genotypes. For example, if there is a specific location where I am not able to infer the genotype with confidence, then I can also consult the reference panel, and there are statistical frameworks that would allow us to use these panels for phasing and imputation. So, but all these solutions use a linear reference genome, which has limitations, because then the limitations of linear reference is that we would face reference bias in polymorphic regions, such as the regions involved with immunogenetic regions. And also, when we have short reads, it can also be challenging to genotype large variants, especially structural variants, as well as variants in repetitive regions, because there both alignments can be inaccurate and even K-man counts can be ambiguous for repetitive regions. So this is a rough overview of how approaches work using a linear reference. And as a result, there have been a number of attempts on extending the genotyping problem for using graph references as well. So in the graph-based reference, you have now a pangenome graph reference, right? Instead of a linear reference. And again, I can think of two kinds of approaches, that is alignment-based and alignment-free methods. So for example, in alignment-based methods, again, I would want to align these reads accurately to these graphs. And based on the alignments, I would infer the genotype at all locus. And similarly, in alignment-free methods, based on the K-mer counts, for example, the K-mer frequency, for example, I can consider the K-mer at this path, what is the frequency of those K-mers in the short reads. Similarly, K-mer frequency of this path, K-mers, and what is the K-mer here, and what is the frequency of those K-mers in the short reads. And based on the K-mer counts, one can also infer the most likely allele that would be part of the target sample. So the key advantage of these pangenome graphs is that both small and large variants can be represented well inside these pangenome graphs. So as you can see as illustration, this illustration is from the Pangeny paper, where you can already see that these variants would appear as bubble-like structures in these graphs. And the problem then is to infer a path in these graphs that would match the allele of the target sample. So given this overview, basically the goal of our work was to develop a rigorous algorithmic framework for genotyping using pangenome graphs. And the motivation here is that most of the existing approaches require complex heuristics for solving this problem because graphs can be very challenging to operate with. So in this attempt to identify the rigorous algorithmic framework, we first start with the assumption that let us say that the target genome is haploid. So we start with a relatively easier problem where we assume that the target genome is haploid, that is ploid equals 1. And now we can think of this genotyping problem as finding an optimal haplotype path in the graph. That is, let us say that's a mosaic of the known haplotypes in the graph, and it agrees with the given short reads. So we will have short read sequence from the target genome. So we would want this optimal haplotype path to agree with the short reads. I'll formally define what I mean by this word agree. And we would want this optimal haplotype path to be a mosaic of the known haplotypes. That is, we allow recombinations. That is, one can switch from some haplotype to other haplotype in the pangenome graph. And this idea is partly motivated by the Lee-Stephens haplotype model and also the Pangini work, which also uses a similar concept. So given this intuition, let me give you a precise problem formulation that we are working with. So let us say we have a pangenome graph which has, let us say, some haplotypes represented as paths in the graph. So for example, you have 3 haplotypes shown here, one in dark blue, one in light blue, and one in green color. We also have reads from the target sample, and let us say from reads we have identified the k-mer matches to the graph. So for example, this substring ACCC is matching to a subpath in this graph spelled by this path AC and CC. So accordingly, you will have a number of k-mers in the short reads, and many of these k-mers would have one or more matches on this pangenome graph. So this is the input, and the optimization problem that we are framing is as following. Given this set of k-mer matches, and let us call them as anchors, we want to determine a haplotype path in the graph with minimum cost, where I'm defining my cost as the number of recombinations, that is, how many times is the path switching between haplotypes. Plus, I would also penalize any anchor paths missed in the inferred haplotype path. That is, let us say that if my haplotype path is not consisting of this k-mer match, then I would penalize for this missing the I would penalize for missing this k-mer match, because that means that my haplotype path is not fully agreeing with the given sequencing reads. So here is an example. Let us say that you have my inferred path is shown in pink color. Then this pink color path has one recombination. So there would be a cost of recombination because I'm switching from this dark blue path to light blue path at this point. And also observe that I'm missing some of the k-mer matches. So for example, this substring CCTT is not matching as a path to this inferred path anymore because I missed this anchor, right? But other anchors I have captured, because you can see that this anchor, the anchor here, and the anchor here, they have been included. But the penalty of this excluding this match would be accounted for. And as a result, the number of recombinations in this case would be 1, and the number of k-mers missed in the inferred path is also 1. So therefore, in this particular example, the cost would be 2. So given this objective function, you want to determine the optimal haplotype path, which minimizes the cost defined here. And the hope is that this optimal haplotype path would be the best representation of the target genome. So let us start with solving this problem and analyzing its computational complexity as well. So when we look at its computational complexity and to decide to see whether this problem is even, do we have a polynomial solution to this problem, or is this problem NP hard? So it turns out that this problem is NP hard, and even if we restrict to binary alphabets. We show this proof by drawing a reduction from the Hamiltonian path problem. So let us say you have a Hamiltonian path instance where you are given an instance with 4 vertices. Now I'm going to convert the instance given to the Hamiltonian path problem into an instance for the haplotype path inference problem. And this I will do by drawing a pangenome graph in which I will create multiple copies of this graph. So for example, if there are 4 vertices in this graph, I'm going to create 4 columns in this pangenome graph. And if there is an edge from U1 to U2, I'm going to put an edge from U1 to U2 across all these columns. So for example, there is an edge from U1 to U2 here, again there is an edge from U1 to U2 here, and there is an edge from U1 to U2 here. So the connectivity of these vertices will be defined based on the connectivity of the original instance. Now, the way we show the reduction is by putting the K-mer matches on this graph. And the way we put the K-mer matches is basically we also have a source and a target vertex here. We will put many number of K-mer matches on the source vertex and large number of K-mer matches on target vertex as well, so that the inferred path is forced to start from source vertex and it must end at target vertex to be optimal. Otherwise, it would lose a lot of anchor matches, which would result in large penalty. Also, we create unique string matches to each of these vertices such that the inferred path is forced to visit each vertex at least once. So for example, if I consider vertex A here, then it should go to T, then it can go to G, and then it must go to C to cover all the string matches. So as a result, such a path would only be possible if there is a Hamiltonian path in the original instance. And through this way, we draw equivalence between the Hamiltonian path problem and the path inference problem that we are trying to solve. So we show the equivalence both ways. That is, if you have an inferred path, then the original instance has a Hamiltonian path. And similarly, if you have the original instance as a Hamiltonian path, then we will have an inferred path with a certain cost. So now through this equivalence, we know that the path inference problem is NP hard. So then what do we do? Should we give up, or should we design some kind of efficient solutions that can still work on practical instances? Well, it turns out that this problem can be solved efficiently using integer programming. And in the next few slides, I'm going to walk you through the integer programming solutions that we have developed to address this problem. And in the end, I will show you that even if the problem is NP hard, we can use these efficient integer programming solutions to scale to problem sizes that are practically relevant. So let me start with the overview of how this integer programming framework works. Okay. So as a first step, what I'm going to do is for the given pangenome graph that we have as input, I'm going to create an expanded graph where I'm going to separate all the haplotypes from each other. So for example, remember that in the pangenome graph, 2 haplotypes can share a common vertex. But in my expanded graph, I'm going to separate all these haplotypes and I'm going to introduce a source and a sink vertex such that I have separate paths for each of these haplotypes. So I'll have one path for haplotype 1, I'll have one path for haplotype 2, and I'll have a third path for haplotype 3. Also, wherever a recombination is possible, for example, at vertex CC, observe that I can switch from haplotype 1 to haplotype 3. So to account for this possibility of recombinations, I would also put these red colored edges to account for the possibility of recombinations. That is, I want to allow recombinations between these haplotypes. Now, moving forward, what I'm going to do is basically I'll introduce the integer programming constraints that would depend on the number of edges and number of vertices in this graph. But one unfortunate drawback of this expanded graph is that the number of recombination edges between adjacent columns can be quadratic in number. That is quadratic in the number of haplotypes. Because as you can see, maybe you may want to switch from every haplotype to every other haplotype. So in that case, you would need to introduce quadratic number of edges, which might later affect the number of constraints as well, because then we will need too many constraints in the integer programming framework that we are going to introduce. So as an optimization, what we are going to do is we will optimize this graph a little bit. And to optimize this, what I'm going to do is the recombination edges, I'm going to reduce them from quadratic to linear. And that the way I do it is I introduce a dummy vertex in the middle of these columns, so that whenever I want to do a recombination, I would go to the dummy vertex first, and from the dummy vertex, I would go to the other haplotype. And this way, the number of recombination edges between adjacent columns remains linear. Okay. So now, once we have this optimized expanded graph, I'm going to start framing my integer programming variables and constraints. So let us say that I will put also the weights on these edges. So I'm going to assume that the weight on these black colored edges is 0, because I'm not facing any recombination penalty for these edges. However, I'm going to put a positive weight on these red colored edges, because this is where I have to put a recombination penalty if my inferred path is going through these red edges. So there I would, for each red edge, I would put a weight of C by 2 if my recombination penalty is C. Because for any recombination, I'll have to take 2 consecutive red edges, which will account for recombination penalty C. So now, okay, so now I hope that this expanded graph definition is clear to you. That is, you have these separate paths for each haplotype, and you have these edges connecting the different haplotypes to account for recombination cost. The black colored edges have weight 0, because there we don't want to penalize any recombination, whereas the red colored edges have a positive weight. Okay, so now I move forward, and just to remind you, the goal is to find an inferred path where we want to minimize the number of recombinations as well as the number of anchor paths missed. So the idea is as following, that is, I'm sorry, just 1 second. Okay, so the idea is as following, that we will use a binary decision variable for each edge of this graph. And I'm going to convert this problem into a network flow like problem, in which that binary variable will be, the value will be 1 if there's a flow of 1 or along that edge, and it will be 0 if there is no flow along that edge. And the network flow analogy comes from the fact that we will try to seek a flow of 1 from the source to the sink vertex, and along the flow, because we have weighted edges, we will check for whether the flow uses the black colored edges or whether the flow uses the red colored edges, and that will determine the number of recombinations in the inferred path. So these binary decision variables will tell us the flow going from source to sink along all the edges of this expanded graph. For example, if my inferred path starts from source to sink in this orange colored manner, then all the edge variables along this path will be 1, whereas all the other edge variables will be 0. And the intuition is that whenever we see this red colored edge, whenever we see a flow of 1 along a red colored edge, that means this is something that we have to minimize, right? Because that's the recombination cost. As well as there is a second constraint, that is we have to also account for these anchor paths missed from the short reads. And we do this by checking whether for each k-mer match, we do not penalize a k-mer match if all the edges in that subpath have a flow of 1, because that means the inferred path is including that k-mer inside it. However, if the inferred path has any edge, so if we have any edge variable which has value 0, then this will be penalized because we are missing this k-mer from the inferred path. So this is the intuition of how this integer programming framework is working. So let me show you how the exact equations look like. So we develop two type of constraints. One is the integer linear programming, in which each constraint is a linear constraint. We also develop integer quadratic programming, in which the constraints can be quadratic in terms of the variables. So let me show you the integer linear programming framework, in which we have an objective function where you can see that I'm minimizing the weight times the edge variables, because the weight is telling me the recombination cost, and the other part is for the anchor matches. So to walk you through these constraints, basically this constraints will look familiar because this is the flow conservation constraint. That is, we have to make sure that the incoming flow minus the outgoing flow should be equal to 1 only if the vertex is the source vertex. It should be minus 1 only if the vertex is the sink vertex. And the incoming flow should be equal to outgoing flow for all other vertices. We have other constraints for the anchor matches. That is, we want to penalize the anchors which are not part of the inferred path. And for that, we put the anchor constraints as well. And in this case, it turns out that the number of string hit constraints, that is the number of anchor hit constraints, is equal to the total number of anchors in the graph. Now, this can sometimes be too many constraints, and that's what we wanted to optimize further. So as a result, we also developed this quadratic programming framework, in which we are allowed to have quadratic constraints as well. And as you can see, this is the quadratic constraint in which two variables are having a product with each other. And this time, again, the same analogy, that is, we have flow constraints, and this time the number of anchor hit constraints are reduced. But the number of anchor hit constraints are now only the number of k-mers in the reads that have a match in the graph. Whereas in the previous case, if a k-mer has, let's say, 5 matches, then we will have 5 different constraints. But now we have only a single constraint for each k-mer in the read. But as you can imagine, this is a trade-off, because now, even though we have reduced the number of constraints, our constraints are more complex, because these are quadratic constraints instead of linear constraints. So we see that there's an interesting trade-off between this linear and quadratic programming frameworks, and we see that one is faster and one uses more runtime when we use Gurobi solutions. Finally, we further optimize this linear programming framework by relaxing these edge variables. We show that even if we relax these two continuous variables, the optimization still returns optimal answers. So this is the overview of how we optimize this integer programming framework so that it can scale to practical instances. So this is how we implemented this integer programming framework in a software called Phi. Phi stands for pangenome-based haplotype inference. The inputs are short reads and pangenome graph. For each haplotype, we will compute these anchors in parallel. Then, to start the integer programming frameworks, we will construct the expanded graph, and we will add the constraints on top of the expanded graph. And once we have the constraints and the variables, we will optimize the integer model with Gurobi. So finally, the Gurobi solution will return us the flow from source to sink that will be used as an inferred path in the pangenome graph. So let us see how this method compares to existing solutions for the genotyping problem. And so let me walk you through the experiments that we did. So this is an integer programming framework. So right now it's not scalable to the entire human genome, but it scales to the specific regions of the human genome. So we focused on the MHC region, which is the polymorphic region on chromosome 6. It is about 5 million base pair in length. And we used publicly available short reads from this MHC region of 5 homozygous human genomes. And we are using homozygous genomes because, as I said, we are still assuming that we are working with haploid genome, where we are looking for a single haplotype path in the pangenome graph. Now we use this homozygous genomes because we have the ground truth assemblies for these 5 cell lines. So using long reads, researchers have assembled these sequences, and we have complete curated sequences for these MHC region of these 5 cell lines. So then we have a ground truth to compare with. So when we infer the path and we genotype a path in the pangenome graph, then we can check we can compare it with the assembled sequence from long reads and see how well it is matching with the complete assembled sequences. The second column shows you the length of these assembled sequences, and you can see roughly these are 5 million base pair long. And this is the statistic of the short read data that we are using. So these are the coverages of the short read datasets that are available publicly. As a pangenome graph, we are using a graph constructed from 49 complete MHC sequences that are available from HPRC consortium. And these 5 haplotypes that are part of the test framework are not part of the pangenome graph. So, because that would imply that the problem would be much easier. So these 5 haplotypes are not part of the pangenome graph. Okay, so I hope this experimental setup is clear now. Let me show you how this method performs in terms of the concordance with the ground truth. And to measure the genotyping accuracy, what I'm going to do is I'm going to check the sequence path that I inferred from the pangenome graph. What is its eddy distance from the ground truth MHC sequence? So that is, I'm comparing two sequences of approximately 5 million bases long, and I'm checking their eddy distance. Lower is better because any distance of 0 means that they're exactly identical, and a higher eddy distance means that there is some discordance between the ground truth and the reconstructed haplotype. So we are using 3 methods: phi, VG, and PanGene. So in PanGene, for example, it produces a VCF file in the end, and we convert that VCF file by incorporating those variants in the reference haplotype so that we get the inferred haplotype as well. So I'm showing you these results for 5 haplotypes that we were using as part of the test framework. And I'm also using different coverage values. That is, what happens with 0.1x coverage, what happens with 0.5x coverage data, up to the complete data. So because we want to evaluate how well this genotyping framework is working at different coverage levels. So these are the results that we find for all these 3 methods. And the key observation is that the proposed implementation is comparable to the existing solutions at high coverage. So for example, here, when we compare against VG personalized pangenome graph construction, then in this case, the results were almost comparable with VG at high coverage values. So you can see a similar values, and remember that the y-axis are in log range. So even small differences in y-axis will be large gaps in the absolute scale. So at high coverage, these methods are roughly comparable, but at low coverage is where we see our method to be competing better with other methods. So phi results in better reconstruction of the haplotype at 0.1x coverage, 0.5x coverage, and even at 1x coverage across all 5 haplotypes. And this may be because we are directly using this Kmer absence presence in our formulation. We are not using any heuristics, and we are not using Kmer counts in our formulation. We are directly using the Kmer presence or absence, because if you think of from low coverage data, other methods are using Kmer counts to predict whether a Kmer is homozygous, heterozygous, and so on, which can be misleading. So, for example, determining whether a Kmer is an error Kmer or whether Kmer is a homozygous or a heterozygous Kmer, that can be difficult with low coverage data. And as a result, that's where we see a significant advantage compared to other methods in haplotype reconstruction. So moving on, we also check, okay, what is the impact when we keep on increasing the pendulum graph size? That is, what if you keep on, if you use a smaller pendulum graph with only 3 haplotypes, all the way to the pendulum graph with 49 haplotypes? What we find is that adding more pendulum graphs helps with the reconstruction accuracy. So you can see that the edit distance keeps on improving as you add more number of haplotypes. However, at the same time, we also find that the runtime and memory usage of our methods are increasing with the increasing number of haplotypes. And this is something that we would like to focus on, is how do we scale this solution to large number of haplotypes going forward. As I mentioned earlier, that we also optimize the integer programming framework by relaxing the edge variables. And here I'm showing you that relaxation helps. For example, in the quadratic programming framework, we get 1.6x speed up, and in linear programming framework, we get 1.12x speed up using this variable relaxation. So all these optimizations that we put in the integer programming ultimately helped us scale to these MHC sequences because we are working with millions of variables and a large number of constraints in this programming framework. So to summarize, I showed you a novel problem formulation for haplotype inference from pangenome graphs, which can be useful for genotyping applications. And this can address genotyping all kinds of variants, for example, SNPs, indels, large variants, and so on. We show that the path inference problem is NP hard through reduction from Hamiltonian path problem. And subsequently, we developed efficient solutions using integer programming that can scale to the regions of interest in human genome. For example, through the experimentally, I showed you that our algorithm can reconstruct MHC sequences with low edit distance from the ground truth sequence. So for example, using only 0.1x coverage, the reconstructed sequence are up to 99.96% identical to the ground truth sequence. And you are welcome to use file. It's freely available on GitHub. As a future work, one obvious thing that we have to do here is to extend this framework to deployed samples as well. And the idea there would be to somehow extend this problem from inferring one flow, like having a flow, ST flow of one. So right now we have a ST flow of one that we are looking at. In the deployed case, we would want a flow of two from source to sink so that we get two paths from the pangenome graph. We also would like to improve scalability to larger graph and larger number of haplotypes. And finally, we are exploring the extension to metagenomics and clinical genomics, where genotyping using low coverage inputs would be useful. With that, let me also acknowledge all the co-authors of this work. So this work was led by Ghanshyam Chandra, a student in my group. And this is an excellent collaboration with Daniel Gibney and his lab, as well as Alexander Dilly and his student, Stephen. So with that, I thank you, and I'm happy to take any questions.
So thank you very much. It was a very nice and very clear talk. I will start with the first question, but I'm inviting everyone else to ask question on the metrics uh, channel. The question is, so you said that the problem is NP hard when the cost is the sum of the number of recombinations and the number of chimers that you're not able to find.
Yes.
Uh, I expect that it is still NP hard even if you consider only the recombinations or only the chimers that are missing.
So if suppose I only consider recombinations, then basically I would output any one of the haplotypes, right? So I would output you, let's say, if there are three haplotypes in the pangenome graph, I would directly output one of the three haplotypes and say their recombinations are zero. Understand what I mean? So I can easily output a path where recombinations are zero.
Mm. Oh, I see, I see, I see, I see, I see. Okay, okay. Thank you very much. And okay, let's see if there is any question. Any more question? And the second question that I have. So you mentioned at the end that you plan to extend it to diploid organisms. What is the added difficulty with respect to the haploid organisms that you have?
So there are two like there's so one key difficulty there is that when we are predicting two paths, uh, my understanding is that the ILP becomes much more complex. That is, it has to do more work. The optimization space, the search space is increasing now because instead of searching one path, it has to search for two paths. So, ILP would take much longer to solve. That is one. But then also from a practical point of view, would have to make sure that like if even if we are producing two haplotype paths giving two haplotype paths to the end user, you would have to see how many switch errors are happening. Like because when you move to deployed genome, uh, when we are giving two haplotype paths, that is we are giving the genotypes as well as the phasing information, and therefore we have to there not only the genotyping accuracy but also the phasing accuracy would matter. Does that make sense? So because when I
Okay.
give two paths, it may happen that the two paths are like switching between each other wherever there is like whenever let's say there is no heterozygous information or there is not enough information to distinguish between the two paths, then it might happen that I can switch between the
Yeah,
haplotypes.
that is very clear. So I see a question in the chat. Thank you for the very nice talk. I missed something in the expanded genome graph concept when you add intermediate vertices to represent recombinations. Could you please explain again the idea and why it enables to scale? I understand somewhere there is the idea to linearize, but I don't get the point.
So if you remember there, if we don't add those intermediate vertices, then we have to introduce all-to-all edges in the worst case. So in the worst case, there may be scenarios where there may be all-to-all connections among the haplotypes, and that's where the number of connections can be quadratic in number of the haplotypes. But when you put a intermediate vertex, then what can happen now? What can I do is all the source vertices which want to recombine, I will connect them to that intermediate vertex, and then I will connect the intermediate vertex to the target haplotypes. So then what happens now is that in the worst case, the number of connections is linear in terms of the number of haplotypes because now all the haplotypes go to the intermediate vertices, and the intermediate vertex then connects to the following haplotypes. So then it's like a all to one and then one to all in the worst case.
Okay. Now since this talk and this discussion is streamed on YouTube, but there is a bit of delay, so we have to wait a
Okay.
few seconds to understand if everything was clear. But I see someone else is preparing another question. So,
So I forgot to say that this paper is up on bioarchive as well. So if people want to read it further, they are welcome to search for the title on online, and there will be a preprint on this.
that's perfect, and that's perfect. Let's see the question that is coming because I see two two people are typing, and then I need them to send the complete question, and then I'm going to. So how this approach would fare with error-prone reads and sometimes shorter than conventional aDNA datasets. What would be the constraints?
So the first question part I understood that if there are errors in the short reads, because we are using publicly available short reads, which also have some level of errors in them. Uh, but the errors will not hurt our, like erroneous schemas will not hurt our formulation, because what will end up happening is that most of those error gamers having errors will not match to the graph at all. So they are not part of the objective formulation. So the optimization problem formulation will basically ignore those gamers, because there's no way we can have the inferred path going including those gamers in uh, the graph. So because if the so this is assuming that the gamer with error has no match in the graph. However, in rare instances, if the gamer with an error has a match in the graph, that's when the errors would hurt. But in practice, what we find is that most of the gamers with errors don't have a match in the graph.
Okay,
The
okay.
second part is this, where we this
Again, so you mentioned aDNA datasets, and what would be the constraint? So if you need to uh, if add more constraints to the ILP formulations, or if you don't expect to be able to manage in practice error rates that are too large, but you have answered that. You have answered that before. So let's see if there is another question.
the errors would also make we would need to keep our gamer length reasonably, like we cannot extend the gamer length too much, because then there's a good chance that every gamer would every gamer would be having error. So that's, yeah, that's one other trade-off that errors would introduce. That if the errors are too many, then we would need to maybe keep the gamer size short.
So one thing that maybe I missed in your presentation, but how long does it take to actually run Groby on the largest instances?
So we, yeah, so we, let me see. I think it takes 2 to 3 hours with all the
So,
optimizations.
so a matter of hours, not days.
Yes, yes, yeah. So
It's very good.
because we have two frameworks. One is quadratic constraints and one is linear constraints.
Yep.
What we see is that the quadratic constraints runs faster. Groby solves the optimization problem when we have quality constraints much faster, but there the memory usage is higher. So it's a runtime memory trade-off that which we see from Groby.
Okay, so I don't see any more questions. So thanks again. It was a very, very nice talk. So
Thank
thanks
you.
again, and we hope to meet everyone in person one of these times.
Thank you. Thank you, Simon. Thank you
Thanks
very much.
again. Thanks
Thank you
again.
very much.